Hello and welcome to It's Worth a Shot, our second webisode of Peers and Pubs. My name is Christina Wright, and I'm the city coordinator for the Boston Cambridge chapter of Taste of Science, but I am also your host tonight. For those of you who are new to our program, Peers and Pubs is a way for any and all of you to learn about some of the work that is written in scientific publications. Each time you join us, we'll be asking a different specialist to explain a scientific publication and bringing you along for the ride. Oh, and we'll teach you a bit about scientific publishing too. Speaking of which, if you were with us during our last show, you should remember that a journal is a place where scientific papers get published. Peers and Pubs is a virtual journal club, meaning we get together after reading a scientific article and discuss it just like scientists do. Didn't do your reading? Don't panic, you'll be just fine. And today's chat will help you if you go check out the paper later. So, what's the difference between a regular journal club and Peers and Pubs? Well, journal clubs are not typically accessible to people outside of science. So consider this your backstage pass. Now, a couple of things before we get started. We have a chat box open in both Zoom and Facebook, so come and say hey. Let us know who you are, where you are, and exactly what you want to know about a COVID-19 vaccine. If you're in Zoom, please leave any questions you want answered in the Q&A box because we'll miss it if you put it in the chat. If you're on Facebook, we'll have someone scouting for your questions, so go ahead and leave them in the chat there. Also, I want to let you know about something we're calling Jargon Alert. Science can be hard to understand. If at any point during the presentation I need clarification, I'm going to hold up one of these signs to get Odelis's attention. Normally, we'd have our guest join me in holding up a sign, but since it's her husband and he's going to be right there, he can just give her a little nudge. Now, why am I telling you this? Because you should feel free to let us know in the Q&A Zoom or chat on Facebook when you need a little bit more information too. To keep things light amongst the COVID fright, we'll have some Halloween-themed trivia after the presentation to see what you've learned. If you want to play, we recommend you have a second device around, like a phone or a tablet, so you can keep watching the stream while you play the trivia. Now, here's the best part. The top 3 winners of our trivia will win some Peers and Pubs prizes, so go ahead and play. It's worth a shot. All right, before we put a COVID vaccine in the hot seat, let's get warmed up with a quick question. If you're on Zoom, go ahead and participate in this poll. If you're on Facebook, tell us your answer in the chat. So the poll is, what comes to mind when you think of the word review? Maybe something job related, like let's hope I get a raise this time around, or maybe something more like Yelp, where are the best eats in town? How about stay back reviewer number 2, or let's hope the experts like my science. All right, I'm seeing some answers coming in now. Awesome. We've got a lot of science-minded people in the audience. So this is great. I see that there's a distribution of all the answers. I'm so glad to have all of you here, and I hope that you learn a little bit more about review in the context of peer review as we go forward. Okay. So for me, as a neuroscientist, the word review is intimately linked with another word, peer. which gives rise to the critical yet sometimes gut-wrenching process of peer review. It's during this process that scientific writing is heavily edited and gone over with a fine-tooth comb by other experts in the field. Before a scientific paper gets published, they can be sent back for minor revisions. Think, this is good, but we need a little more salt. Or for major revisions, think, this could be good, but it's supposed to be a turkey dinner and you forgot the turkey. Or it could also be rejected, like burnt toast. From there, you can try and submit it somewhere else that's a better fit because all journals aren't the same, or you can go back to the lab and get more data. Anyway, peer review is how scientific papers, like the one we're discussing today, become published in a scientific journal. All right, enough with the extras and on to the main event. Please let me introduce our scientist and presenter, Dr. Odaelys Pollard. She is a native of St. Croix in the US Virgin Islands who left the tropics and migrated to the Northeast to become a microbiologist and an immunologist. She is currently the scientist and educator at ROC EDU, Rockefeller University's Science Outreach Program, where she interacts with the K-12 community, teachers, and scientists. In her spare time, she enjoys trying out new recipes, playing with her 1-year-old, and watching movies with her husband. Speaking of which, she's joined today by her husband, Teyvon, a New York City MTA train engineer and operator. He is a movie lover, born and raised in Brooklyn, New York. Everyone, please welcome Odelis and Teyvon. Hello.
Hi.
All right, so because we have a husband and wife combo on here, I'm going to start us off by asking, how did you meet?
You wanna?
We met on Tinder.
Get right to it.
Perfect, awesome. No, I know, I'm on the dating apps. Maybe I'll find my husband that way someday. Fingers crossed. All right, awesome. So, well, we're glad that you're together,
Yeah.
and we're glad that you're here today. Teyvon, I have a question for you.
Mm-hmm.
Has your concept of what a scientist is like changed since you met Odelis?
Not really. Well, I personally never thought of a, some people think of a scientist as someone like, some crazy scientist, Doc from Back to the Future. But I've never thought of a scientist as being, what is portrayed in the media. So when I found out that was her profession, that's what she loved to do, I wasn't really surprised because, there's a lot of people are into a lot of different things, and I don't judge or profile or stereotype any. So
good for you. Incredible. All right. So now, Teyvon, how much do you know about how vaccines are made?
I know very little, probably the bare minimum. I just know that vaccine, in what I've been taught to believe is a vaccine is basically them giving you the virus to make your body fight it off.
Sounds about right.
Yeah.
Yeah, so this is not my area of expertise either. So I'm really curious to see what Odelis has in store for us today. Now, one more question for you, Teyvon. How much do you know about scientific publishing?
Nothing at all.
All right. So hopefully we got to teach you what a review in the context of peer review is today. And we're going to have more familiarity with the publishing process just by going through a scientific paper with Odelis. All right. So before we move forward, I just want to say to our audience, I want you guys to remember to leave your questions about the presentation or your jargon alerts in the Q&A box on Zoom or in the chat on Facebook, and we'll get Odelis to answer your questions later on in the program. And all right, I guess that's it. Let's talk science. Odelis, I'm going to hand it over to you.
Awesome. So let me share my screen. Let's see if technology works with me today.
Fingers crossed.
Yay. Awesome.
Perfect.
Oh, so welcome everyone, and I'm super excited to have you all on to learn a little bit more and to listen to me explain to Teyvon and you out there exactly what it is when we say COVID-19 vaccine. And so the paper I'm going to talk about today is entitled COVID-19 Vaccine BNT162B1 Elicits Human Antibody and Th1 T-cell Responses. So pretty much all that really means is human immune response to a COVID-19 potential vaccine. But I'll try to break it down a little bit more in this presentation. So like she mentioned, I'm with Rbd Science Outreach. This is my husband, Teyvon, who knows the bare minimum of vaccines. So I'm hoping to teach everyone a little something today.
I'm ready to learn.
All right. So what do you know about coronaviruses?
So coronavirus is like the common cold. That's the main thing I've
Yeah, so the common cold. So many of us may have had a coronavirus some time ago. So the human coronavirus is one of like the more popular or well-known type of coronavirus. What else? Do you know anything else about coronavirus?
Aren't there a bunch of different ones?
Yeah, so there are actually many different types of coronavirus. There's like the alpha coronaviruses and beta coronaviruses, which are usually the ones that cause like coronavirus infection in humans, so human coronaviruses. But many people may have also heard of MERS and SARS, right? So we had an outbreak, I think in 2003, 2012, maybe. So it was like the Middle Eastern respiratory syndrome and then the severe acute respiratory syndrome that occurred. But no one really cared about it. You know, you get a cold, you get over it until 2019, when everyone started hearing about this thing that occurred in Wuhan, China. And then all of a sudden in March, we were in the midst of a global pandemic. So here we're going to talk about specifically SARS-CoV-2, and it's named SARS-CoV-2 because it's closely related to SARS-CoV-1, which was responsible for the SARS outbreak back in, I think it was 2012 or so. All right, so cool.
Got it.
All right, so what do you know about viruses?
in what?
Like just viruses in general.
Well, I
You
know
can catch them.
different types of viruses.
Different types of viruses.
There's viruses you can have that make you sneeze, and there's viruses that do other things.
Yeah, right. And then some people are like, oh, are viruses alive or are they dead? Right. And so by definition, scientists has kind of come to a consensus that viruses are non-living, but they do have genetic material. So here I'm going to show you, I'm showing you, we have the SARS-CoV-2 virus. And the virus is a pretty simple molecule, even though it like causes like mass injury all the time. And so here's the virus, and inside the virus is the genetic material. So you know about DNA, you know about RNA, right? So there are viruses that actually have DNA as its like genetic information, and some viruses that have RNA. For SARS-CoV-2, it has RNA as its genetic information. But RNA needs to be protected, just as our DNA is protected in our nucleus within our cells. And so in order to protect that genetic material, we have this fat layer around it. We call it a lipid bilayer. Lipid and fats, pretty much the same thing. All right, and then on the surface of it are kind of like the more important ones. So like the proteins. So proteins are like the functioning components in your body. And that gives you like a specific characteristics, and it kind of does a lot of the work in your body and in your cells. In our case, the major protein for coronavirus is called the spike proteins. Do you know what the spike protein is used for?
No.
Okay, cool. So the spike protein does several things. So in order for a virus to enter your body, it actually has to bind and recognize another protein in order to enter in, right? Sometimes they can like just be engulfed into the cells, but a lot of times they have proteins that can bind to other proteins in your cell. For SARS-CoV-2, we have this spike protein that binds to a protein on the human cells called human ACE2 receptor. And that binding actually allows for SARS-CoV-2 to enter the cell. And once the virus is inside of your cell, it can replicate and make more of itself. So that's why it needs the whole cell, right? So it releases its genetic information, it like hijacks our machinery and it makes more of itself. And then our body recognizes it as something foreign, and then it produces an immune response, usually antibodies from B cells, or it can use like a T cell response. All right. You good with that? All right, so now we know how virus enters our cell, more specifically SARS-CoV-2, and we know that it elicits some type of immune response. But what do you know about the immune system?
Our immune system fights off anything trying to attack the body.
Yeah, right? And so what do you think people do in order to hijack that and make it into like a vaccine? So how would a vaccine fight off a virus?
Well, like I said, they basically inject you with some form of the virus, so your immune system could respond to it.
Yeah, so you know, pretty decent amount. All right, so that's pretty much how it works, right? And so in our case, if we look at SARS-CoV-2, right, scientists work really hard to understand how viruses work. We try to understand what the proteins are that are responsible for it to enter our cells, what the proteins are that are responsible for eliciting this immune response. But they also study how it replicates in the cell. And they do that for many different viruses as well as other microbes. And so once we know what part of that virus causes that immune response, we can actually study that even more. So in the sense of coronavirus, scientists are really interested in this spike protein, because that spike protein is actually what's mostly responsible for recruiting what we call these immune cells or antibodies to fight off the infection. Now this is a very simplified version, but our immune system has a lot of different cells that do a lot of different things. For this paper, I'm just going to focus on antibodies, which are these proteins that are produced by the immune system. And we have like 5 different types of antibodies. But the more important one for today and for like long-term immunity, we think about IgG. All right, so IgG antibodies are kind of what we look at to indicate if someone is like protected or immune from a new infection. And so what happens is that when your body is introduced by the actual virus or part of the virus, all right, it'll produce these antibodies that's specific to that virus or parts of the virus. And so what happens is whenever you come in contact with that virus, these antibodies are primed and they know, hey, I recognize that thing, all right, let me go recruit some of my other friends. I'm going to capture that virus. And it can do couple things. It can either bind to that virus and call its other friends to be like, hey, help me get rid of this thing. Or it can just all cover the virus in terms of like all the antibodies covering the virus and just prevents it from infecting your cells. Right? So it's pretty clever that scientists have studied how the immune system works. They've studied how viruses work. And now we can hijack part of the virus to mount an immune response without infecting us with the real virus. Good.
Super cool.
Awesome. So there are many different types of viruses, vaccine candidates. And so you've been vaccinated before.
Yes.
How many vaccines have you had?
I've had vaccines I don't even know what I was taking. In the military, they just put you in a line, you walk down the line, and you just get constantly shot in your arm with the needle.
Right, and so you probably got the vaccine for the flu, right? Which we're gonna get eventually. Hopefully, like this week, we'll get the flu vaccine. We haven't done that yet, right? But they have vaccines for like other types of viruses, and these vaccines are made in many different ways. I just want to focus in on coronavirus vaccine candidates. It's a really neat figure from Nature. So Nature is a journal where we actually got this paper from, and they do a really good job of reviewing what the vaccines are that are in production. And so you can have, like you said, the virus or some part of the virus. So scientists can either kill the virus or inactivate the virus and then inject it in you, and then you mount an immune response. They can actually use the genetic information of the virus, right? Inject that, and then your cells would make the protein, and then your body would recognize that, and then it elicit an immune response. They can have like another type of virus and put the actual virus that they want the immune response against inside of it and inject that. Or they can just inject you with the proteins that are responsible for mounting an immune response in your body. So I say all that to say that there are many different types, But the type that this paper talked about is a nucleic acid vaccine. So nucleic acid can be either DNA or RNA. In this particular paper, they're using
Sorry. Wait, so with the nucleic acid one, you said if your body is making the protein. So is that spike protein that you were talking about?
Cool. So they actually, so they'll so we have all these type of bioinformatics tools where we can actually see what the genes are that can translate into that protein, that spike protein. So the RNA would be that sequence that's going to translate into that specific spike protein.
Wow. So it's almost like your body is like creating the vaccine and then mounting the immune response.
Yeah. So it's just taking the information. It's like, oh, I see this information floating around. You know, your immune cells either present that as the protein and then recruits other cells. So it's really fascinating how like our bodies can take up these different things. It doesn't really pick and choose like what it is. It just know that it has to do this job.
Awesome.
So in this paper, they looked at an RNA vaccine. And so RNAs are fairly unstable. So, and we also need a way for it to get inside of the cell. So what they do, they actually enclose the piece of RNA that encodes for the spike protein in coronavirus's case in a lipid coat, like a fat coat, right? And they do that because the cells in our body are surrounded by fats. We call it a phospholipid bilayer, but it's pretty much mostly fats. All right. And so what happens is that these two lipid membranes can fuse together. The RNA can then enter the cells. Proteins are made. Your immune cells can pick up these proteins, present them on their surface, and then recruit these like antibodies and all these other immune cells to come and fight the infection. Right. So pretty cool. Now, what's really cool, well, I thought it was cool about this vaccine, it's part of what they call a Project Lightspeed. And so, you know, this pandemic started, I think they announced a global pandemic in March or February of this year, right? And it's killed over 200,000 people, infected millions worldwide. And so we're really like pushing hard to bring about a vaccine to hopefully infer immunity across the population. And so this RNA vaccine was one of many that they were looking at. And so this one is BNT, and I think the BNT stands for BioNTech, because the company that actually produces it. And we just like put nomenclature to keep track of like all the different types of vaccines that we have. And so here for SARS-CoV-2, remember I told you the spike protein is what's responsible. They're actually a small portion of that spike protein that we know is needed to mount an immune response. So you can either make genetic information to encode for the whole spike protein, or you can use that genetic information to encode for just that important part of that spike protein. And in this paper, they're encoding for that important part, and it's the Rbd domain. All right, so they made this mRNA and it enclosed it in a lipid nanoparticle, or just like a lipid coat, in order for it to fuse into the cells. Now, the RNA vaccines, a lot of people haven't really heard about it because there are actually no RNA vaccines that are out for public use, right? So there are many pros and cons of using an mRNA vaccine. And so many different pros. So it doesn't integrate into your genome, and not that there are vaccines that can integrate in your genome, but when you think of like theory and, you know, when you're thinking about like what may have happened, DNA can maybe have a likelihood of integrating into your DNA, but you won't have that problem with mRNA. Right, it's well defined. They've done it in preclinical trials and clinical trials. So preclinical trials are the trials that you do like in the lab and in animal models before you do it in humans, right? And it's been safe in a lot of the clinical trials in humans that they've done. You can reproduce it really fast. It's really scalable, so you can make lots of it in a really short time. So like I mentioned, the con about it is that there are currently no approved vaccines that are mRNA vaccines. And because it's only, you know, telling your body or trying to mount an immune response only for a small part of the vaccine, you might get a lower response in comparison to, you know, giving your body the entire killed portion of the vaccine. It kind of makes sense, right? Cool. So let's talk about this RNA vaccine and what did they do in this paper. We're going to get into like the nitty gritty, the good stuff. All right. So this paper was a phase 1, phase 2. Do you know what those mean? Phase 1, phase 2, phase 3. Have you ever heard those terms in your life?
I've heard those terms, but not in this
context. All right. So the FDA does a really great job of, you know, looking at how effective this vaccine is, how safe this vaccine is. And so it has to go through 3 different phases before it's approved for use. And so the first phase is that you have a small cohort of volunteers and they have to be healthy volunteers. And you're looking at several things. You're looking at, is this vaccine safe? All right, do they seem to work? Are there any side effects? What are those side effects? Once it passes that phase, we've like tested it on 50 people, it seems to be super safe. Then you can move on to phase 2, where you're just increasing the amount of volunteers to make sure that you're still seeing those same results that you saw in phase 1. Mm-hmm. And once you pass phase 1 and phase 2, then you can go on to phase 3 and then take in several thousand people, right, to test this vaccine. And you want to make sure that they're having immune responses to it, right? Because what's the sense of getting a vaccine if your body isn't mounting an immune response? Mm-hmm. And also making sure that all these thousands of people are not having any adverse effects. And so what's really cool is that there are many vaccines now that are in this phase 3 trial. But this paper was published, I want to say like sometime in August, it was accepted in August. And so this paper goes over the phase 1 and phase 2 part of the vaccine. All right, so there's many ways that you can do a clinical trial. So this clinical trial was non-randomized, right? Mm-hmm. No, non-random, so it's not random, right? So they have specific sets of people in specific groups. It's open label, so the participants actually know what they're getting, and the scientists or the people who are given the vaccines know what they're giving. And it's a phase 1, phase 2 study, which I just explained. And so in this study, they had 60 participants, and they were all 18 to 55 years old. And so with those 60 participants, they had 12 in different dosage groups. And so they had 12 who were given 1 microgram of the vaccine. There were 12 that were given 10 micrograms, 30 micrograms, 50 micrograms, and 60 micrograms. All right, now this study was done in Germany. And there was actually a very similar study done in the US with about the same number of participants, but they only did 10, 30, and they also did 100 micrograms. So they're kind of just trying to see if they can reproduce a lot of the same results that they saw in another country. Now, they did several things with these participants. The ones that had 1 to 50 micrograms, they did a prime and a boost, and I'll talk about what that means in a second. And then for the 60 micrograms, they did no boost. So what does prime and boost mean? And so here is a schedule of how they did the vaccination. So prime is the first time that you're given that vaccine. Mm-hmm. All right. Have you ever had like the tetanus shot? Yeah. All right. And have you had it more than once? Yeah. Yeah. All right. So a lot of times they'll give you the tetanus shot and they'll give you like another one to boost your immune response to that microbe. And so here they're given a prime on day 1, but they're also collecting their antibodies on these specific days. Right. And then you have the boost on day 22 to try to mount a higher immune response. And they're also taking antibodies in that day, as well as 7 days after boost and 21 days after boost. Right. So prime is the 1 shot, boost is the 2nd shot that they're getting. All right, so let's see what they saw. So can you explain this graph to me?
Well, this is the number of subjects in the days, and each one is the amount of the vaccine that they gave them.
Oh, you're good. Right. So the first thing they looked at is how safe and how tolerable this vaccine is. Mm-hmm. And they looked at like the injection site, so the local events. And so what this slide is showing, like you just said, on one axis, you have the number of subjects, then you have the days after the vaccine and the amount of vaccine that they gave them. And they rated it based on these parameters. So they had no events, they were mild, moderate, or severe. Now here for pain, what they saw was that there were a couple people that had severe pain at the site, but it seems, oh, Jargon Alert, sorry.
No, you're fine. I just have a quick question that's actually in from our audience. How do they decide what like acceptable side effects are for like?
So it's also based on side effects that we expect from previous vaccines. So like vaccines that have been approved. So like if you've ever had your flu shot, right, you realize like it's sore at the site for like a couple days or two. And so they know like that's like a normal side effect. This vaccine has been approved. It's been distributed to many people. So they kind of do it based on like previous vaccines that they've had out there.
Gotcha.
Cool, and like
thanks.
What they think is horrible. Yeah.
Yeah.
And so what you see is that when you increase the amount of vaccine, what happens?
You increase the amount of pain.
Yeah, right. And so it seems that at 10 micrograms, not many people felt pain, right? But the higher you go, there were, a couple in those groups that experienced pain on the first day, and it seems like on the boost of the vaccine. And so they looked at several other things. They also look at swelling and redness. I'm just showing pain, but the others were less severe than the pain. So now this shows a very similar thing. Instead of looking at the local effects, we're looking at systemic effects. So things like headaches, chills, fever, diarrhea, vomiting. And so fatigue was one of those that they saw among the most participants. And so you can see the same thing occur where you have higher amounts of vaccine, you have more fatigue, right? Very, very few people witnessed vomiting. Cool. So would you, based on like the side effects, would you consider taking this vaccine at like the low dose?
Yeah, I'll take it. Everybody gets a little pain when you get a needle.
Yeah, right.
A little bit of fatigue. I'm always fatigued. I'm always working. So that's nothing.
All right. So what did I tell you? Like, what did you get from that first section? Like, well, how would you summarize it?
The way I would summarize it is that the higher the dosage, the more side effects that the individuals experience.
Yeah. Right. So it's dose dependent, right? Mm-hmm. So the higher amount of vaccine that you give, it seems that you have more side effects. And the side effects also seem to increase with the second dose. So after their boost shot.
Cool. I actually had another quick question. Yeah. Why do you think they didn't include people who are over the age of 55? Because people who are over the age of 55 also get the virus, right?
Yeah, so they actually went ahead and amended it for the next study that they're doing, the phase 3, where they can include patients over 55. So that was also a concern from other people because we know they are the vulnerable group. And so they actually went ahead and, changed the study parameters to include those patients.
Yeah. Awesome, cool, thanks.
No problem. All right, so what does this look like to you? Another beautiful graph.
I see some Christmas trees.
Oh, can't wait for Christmas.
This one needs more explanation than the last one.
It's a lot more targeted here. All right. So again, let's look at the y-axis. So here we have a log scale. It's just increased by the power of 10. So like here is 100, 1,000, or 10, 100, 1,000, and so on and so forth. But this is also showing the amount of IgG. So those antibodies that I showed you. And they did it in units per mil. Mil is like a volume. So the way that they measure this antibody. On the bottom, the x-axis, we have the days after initial vaccination. So pre is they took analysis before they got the shot, right? 8 was 8 days after their prime. And then 22 days after their prime. And then 29 days were 7 days after the boost. And this was 21 days after the boost. All right, so we're looking at what are the antibodies looking at before I gave the vaccine, right? 7 and 21 days after I give them the vaccine, and 7 and 21 days after I give them the boost, right? And then on the right, we have the amount that they're giving them. right? Or what the labels are. So we have the the amount of vaccines that they gave, and HCS is actually, they looked at people who've been infected with COVID-19, and you wanted to compare how their antibody responses look with like the vaccine antibody response. And what they saw was pretty decent, right? Oh, we're also looking at Rbd specific IgG, right? So that's that binding site on the spike protein that this vaccine is specific to. So in the first one, we're looking at 1 micrograms of the vaccine. And you can see 7 days after the vaccine, you see a little bit of response. This is like the mean of the amount of IgG that they see. 21 days after the vaccine, and this is before they did the boost, they got about 265 units per mil. Then you see after the boost 7 days, it went up a whole like power of 10. So it went from 200 to 2000. And then 21 days after, it went up like another almost 2000. All right, so pretty good. Now, when we look at all the other ones, we're seeing that similar trend, and we could even get up to 25,000 7 days after the boost if we did a 50 microgram boost of the vaccine. Here we have 17,000, so almost to 20,000. But I think what's even more important, and I think a little bit more cool, is that when you look at the patients who actually were previously infected, you get a higher response even at the lowest dose than those patients. So after you get a boost, you get 2,000 units per mil of IgG in comparison to an average of 600 for those who've been previously infected. So it almost seems like the vaccine is more effective at mounting an immune response than people who are actually infected with the virus, which is really cool for if we think about like protection.
That's crazy.
Right? I thought so too. All right, so let's summarize it. What did we see about the antibodies based on what I showed you?
Based on what you showed me is one thing I had a question about is the antibody response from the vaccine being higher than if you were to actually be infected with the disease. Is that really a good thing that what we're injecting each other with causes a higher response than the actual
I think
virus
so,
itself?
Because like when you get in the vaccine, you're not sick. Like if you were symptomatic and you had to actually get COVID, like you're way worse off than getting the vaccine, and your body is actually producing more of an antibody response than you would. But also to note is that these patients were only infected once, right? And with the vaccine, we're giving them like a second dose. So we're like priming them, saying the immune system, like, hey, let's build up some antibodies, and you're priming the immune system, or you're giving it a boost the second time, like, hey, produce more antibodies. And so the thought is that, there's a lot of talk about like, how long we have immunity for if we've been infected. And so we're hoping if we can get a vaccine that can offer a longer immunity than if you were just infected with the virus, it'll be better off for us.
Okay.
Good question. All right, so pretty much I showed that this vaccine produces this Rbd specific IgG. And it also, I didn't show it, produces neutralizing antibodies. So there are antibodies that can bind the virus, and there are antibodies that can like neutralize it and prevent it from infecting your cells.
Does it like blow it up?
Actually, I don't know, blow it up. I guess there's like T cell responses where they like attack and like degrade it. So I guess it is kind of like blowing it up. I would like to see like an electron micrograph of like that happen. And I'm pretty sure there might be something out there of like
That'd
a
be pretty
virus
cool.
dying. Yeah.
I'm just imagining like these immune cells going like choo choo choo.
And it's just like yeah. And so we saw that even if they gave a dose of 1 microgram, you can actually mount a pretty good antibody response. And it was actually more than people who've actually been infected with COVID-19. So I think overall, I mean, what do you think? Because I'm a scientist, so I might have a little bit of bias.
That's a good thing. It's making our bodies, we're evolving. Fighting off more, the new viruses come along. We build up the high number of antibody responses.
Yeah, right.
And continue to fight off these things that come at us.
Yeah, so we need scientists.
Of course.
Yay, science. All right, so I just want to give you like a brief overview. I think think you kind of understood what I said, right? And so there's some main points I just want to point out. So one, people who got this vaccine, the one that ended in one, reported no serious adverse events. 2 doses produces a pretty strong antibody response. And the results that they saw were actually very similar to the US study, which is pretty good. Now, there are some downsides. What we have to keep in mind that this clinical study where it was only 60 participants, right? And like y'all mentioned, like there were no patients over 55, and we know that those are the vulnerable group. And they, you know, made that right with their next study. And so we still don't really know what long-term immunity looks like with the vaccine. So I think in this study, they like looked at it up to 43 days, and they actually saw that there were still some antibodies being produced. And their goal is to look at them up to 6 months afterwards. And so to see like how much, you know, the antibodies are going down. They have been a couple of studies looking at people who were infected like in March, and they looked at their antibodies 6 months later, and they do see a decrease in antibodies. So we're hoping that the vaccines that we're given can keep your body producing these antibodies, and these antibodies can stay around to fight off the infection.
Did they also have an equal number as far as say men to women and by ethnicity and race? Because, you know, the virus affecting women more than men.
Oh, men more than
women, and it's affecting certain races more than others.
Yeah, that's a good question. So I looked into that. And for the US study, they had 1 African-American person out of, I think it's about like 50, 60. In the Germany study, they also just had 1 African-American person in the study. Now, there's so many things to go with that. I think they had about 50% women and men. They had women who were healthy, and they were all healthy participants. I think they realized that as they were doing the studies, and I just saw, I think it came out like a few days ago. So Moderna actually has a vaccine out, also an RNA vaccine out in phase 3, and they have 30% minority population in their study. So they went really hard on bringing in people on board to be a part of the study because they knew exactly what you were saying. There's some people who are more affected by the virus than others.
That's awesome.
Yeah. And so even though the results from this vaccine looked pretty good, the BNT162b2 was the vaccine that actually went to phase 3. And so the difference between the 1 and the 2 is that the 2 encoded for the entire spike protein. So it actually elicited an even stronger response in your body in comparison to the 1. And for some reason, it actually had less adverse events than the B1. So that was the one that they approved to move on to phase 3. So now we have thousands of people that are in these phase 3 trials. I'm thinking, I don't know how long it's going to take. I guess it just depends on like people who are actually, you know, participating and like the approval process for the FDA, which is, you know, going really quickly because of our current pandemic. But hopefully in a few years, we'll have like, this would be like a memory, a distant memory.
That would be lovely.
Awesome. So yeah, so that's it. Any questions?
No, I haven't. You explained it very well.
Oh, thank you.
Yes, you absolutely did. It's really an incredible topic, and I'm so grateful that you took the time to share that presentation with us. Thank you so much. Sifting through all of the talk about vaccines and COVID-19 in general has been a challenge for me, and I'm sure a lot of other people. There's just a lot of information out there, and having the opportunity to walk through a paper like this with somebody like you is super awesome. Okay, so thank you to both of you. Before we get to our trivia and Q&A, I want to let all of our viewers, remind all of our viewers at home that Taste of Science is a nonprofit that relies on science lovers just like you to keep our programs running. If you could go to tasteofscience.org and hit donate, we would be so grateful. With that, let's get to our trivia intermission. We're gonna sit back for 5 minutes on this end, play some music, and see how all of you at home do. So go ahead and head to the chat box for information on how to get into the trivia game. You can click the nearpod.com trivia link there, or you can go to www.nearpod.com, that's near with an N, pod with a P, and type in the code W84TK, which is also going to be in the chat. When you enter your name for the trivia, feel free to put a spooky spin on it. I don't know, add a bunch of O's and a little boo in your name somewhere. But for prize purposes, remember, we do have prizes for the top 3 winners. Make sure we can identify you from your name, and make sure that we can see that it matches whatever you put on your registration information for the Zoom webinar today. Last thing, please note, if you're on Facebook, you'll want to watch your device and not the live stream during trivia, because there will be a bit of a lag. Right, I guess, anyway, enjoy, good luck, and we'll be back in 5 minutes. All right, everybody, thank you for playing the trivia and welcome back. Congratulations to Dom, Leslie, and the Great Pumpkin, I think, if I have that right. If you are the 3 winners, don't forget to reach out to us via private message through Zoom or one of our social media channels, to make sure that we can get you your prize. All right, so now let's move on to our question and answer portion of tonight's program. We're gonna go back over to chatting with Odelis and Teyvon. We have so many questions from our audience, so let's get started. Question 1, we have Leslie from Zoom wants to know, a vaccine has to be about 50% effective for approval. How is that going to be very useful? So I think she's questioning whether 50% is enough.
That's good enough, yeah. So that's a good question. So I think they did that number because of the speed of trying to develop the vaccine, and that's 50% in comparison to the placebo. So you're looking at effectiveness of the vaccine versus people who didn't get the vaccine. When we compare to like the seasonal flu, I think the effectiveness of the seasonal flu vaccine is about 20 to 60% or so.
Wow.
And so it's almost like the risk to benefit ratio. So it's better to have 50% that are immune, thinking about like if we still have to take some of the safety measures that we're doing right now, going forward. But I think a lot of scientists are aiming for like the 70%, 60% for the vaccines to become approved. So yeah, it's not a perfect number. There's like no vaccine that's 100%. I think some of the most effective vaccines are like the childhood ones that we take, like the MMR. And things like that. Yep. But yeah, it's really difficult to have like a vaccine that's 100% effective. But I think scientists are really working hard to get above that 50%.
Yeah, absolutely. It seems like it should be so easy, right?
Yeah, no, it's not. Like viruses are super smart. And it's kind of scary. I feel like there are scary movies about viruses that like take over. But yeah, I mean, we could just do as much as we can to combat them in the moment. And just hope it works.
Yeah, absolutely.
It's a sad reality, but you can do the best that you can, but you can't save them all, as they say.
Yeah, we have to try our best to save as many as we can. Yeah.
Definitely. You mentioned viruses are pretty smart. I think that gets at our second question, or leads us over to our second question pretty nicely. So Barton from Zoom wants to know, how would the COVID-19 virus mutations impact the efficacy of current vaccines that are being tested?
Yeah, so the good news is there were some studies that were done that show that it doesn't seem to mutate quickly. And then mutation, we talk like everything mutates, like things happen. I think the things that's concerning is mutations that actually would make a difference in terms of infectivity. And like, what do you call it? That make you more sick? I can't remember the word. So like how well it can infect you and how sick it can get you. And so when we're thinking about mutations, one thing that they did do in the study, they used many of the variants that they had access to of that like spike protein important part. And it showed that the antibodies that they produce was still effective at neutralizing those viruses. And so even if mutations occur, it might be less likely for it to occur in a manner that makes it worse, because it seems to be pretty effective right now. And so I'm hoping that, like it can happen, right? But studies have shown that it's really unlikely to mutate to that effect. And hopefully the antibodies that we're producing, I think that's also a reason why they're doing the spike protein vaccine instead of the small portion, so that they have like more coverage. So if a mutation was to take place, it's not gonna like completely outdo the entire spike protein. There might be like few amino acids that may have changed, but hopefully the antibodies are still effective at finding and binding them.
Cool. Awesome. All right. Our next question comes from Keith over on Zoom. And Keith wants to know, how do the side effects seen in the treatment groups compare to the control groups? Were any of these effects seen in controls?
Yeah, that's a good question. I actually did not go through the paper. So this particular study did not have a placebo group. So everyone got the vaccine. I know like some of the initial studies did have a placebo group, and I don't think any of them had, no, I lied. I think some of them had the pain, like the local adverse events, but I can't remember off the top of my head. I'll have to go back and check, but I don't remember seeing any of like the systemic events that they saw.
Yeah, and that would make sense because you're still getting a shot, even if you're getting the placebo, and it'll still hurt a little bit, I'm guessing. All right, cool. So up next, we have Carrie from Zoom who wants to know, does this paper mean that we all have to get 2 shots because the boost was better?
It's looking like a boost may be needed for the vaccine. Now, what I don't know is how they'll schedule the boost. So I don't know if it'll be like a vaccine now and then 2 weeks later you have to get the boost. I mean, but also like vaccines take many iterations. So like if this vaccine gets approved, we're not gonna just stop trying to improve the vaccines. Like even the flu vaccine, when we think of it, there are many iterations of the flu vaccine. They have like the Flonase that came out. They're always trying to refine and make sure that it's at its best and optimize it. So yeah, to remember the rest of what I was gonna say, but I think that
that's fine.
I think that answers the question.
Yeah, absolutely. No, you totally did. The next question we have is an anonymous question, but they were specifically asking about all the circles on like slide 33 towards the end of your presentation. I think it may have been like the Christmas tree triangles.
Oh yeah, the Christmas tree. Yeah, so the circles just indicated like what the label was. So there were circles so they're like the individuals. And so the number is the mean. So like they took the average of all of the IgG amounts that they got, and the actual dots were the individual amounts. And so they're just showing you like how much they spread on that curve. So like in that cohort, you might have one that was as low as 1,000 units, right? But they also had someone who had like 5,000 units. But the average was, I don't know, like 3,000 units. So they're just showing you the individual response, and the number was the average.
So each dot is a person.
Mm-hmm.
Got it. All right. We have another anonymous question. They want to know, what about long-term side effects of vaccine? Is there any way to predict them?
No. I mean, yeah, and that's one of the caveats with anything in terms of like treatments or vaccines that are developed. I mean, you do your best to ensure that it's safe and effective in the time frame that you're doing it. A lot of scientists are doing prolonged studies where you're continuously looking at like the patients that you've, gave the vaccine to. And I think they may be doing like follow-up with the people that they've done like in the phase 2, maybe the phase 3. So yeah, it's really hard to tell. I mean, you're also going based off of like previous vaccines that you've done. And like I said, the mRNA one is like I would say novel in terms of like this would be the first to be distributed for human use. But I think based on preclinical and clinical trials, there's no nothing that's alarming scientists right now in terms of if long-term effects would happen. But that's a really hard question to come at. And I don't think we know that answer.
Yeah, that makes sense. But I feel like all in all, we've developed a lot of vaccines. So like we have people who have a lot of information behind them working on these new developments. So that's comforting. I think.
So thinking about like all the vaccine we've had over time, and it seems like, one thing I like to think of as well is like when you think of like the lifespan prior to vaccinations, right, and the lifespan that we have now, there are other things that may have played a role, but thinking about like we're able to extend our lifespan because we're able to manipulate I guess how things work to make sure we can try
the
to
natural order of things. Yeah, for sure. All right, we have another question. Leslie from Zoom wants to know, weren't all 3 trial stoppages related to neurological issues, even though they were from 3 different trials? The similar reasons seem a bit unsettling to me. Do you have any thoughts on this?
Yeah, so there are many trials that are happening. I did hear about a couple that were stopped, or a few that were stopped because of issues. And they restarted the trials because it seemed to be like a really rare response that happened, and it was based on like underlying event in that patient. I think which is also really important for us to expand the amount of people that we have in terms of like the different types of people, different ways of living. Unfortunately, we could usually most times when we're doing like these clinical trials, use healthy volunteers. And so like as you extend the study, you can incorporate more people, but it's a risk to benefit ratio, right? There's always people who may have like an allergic reaction, who may not respond well, and you just have to figure out like what that number is. And I would love for us to say 100% of everyone who will get the vaccine would, nothing would happen, right? But that's unrealistic to say.
Yeah, absolutely. That's crazy. I've never even thought about, this is a really naive thing to say, but I've never thought about not being able to take a vaccine because of like underlying health conditions or something like that. So that's a whole nother layer.
Yeah. And so that's why herd immunity is such a big thing. I think that word has been going around a lot, where a lot of people is like, oh, I'm young and, I don't need the vaccine because, if I become sick, I'll be fine. But you have to think about like, if you protect yourself, you protect the vulnerable that's around you. And I think that's the case now with just COVID-19, like where people are like, oh, I'm fine. I'm like 20 years old. I can, if I get COVID, I could just wake up the next day and carry on with my life. But you don't know who you're interacting with, right? And I would hate to be that person to put another person at risk because, I didn't take it seriously or.
Absolutely. Yeah, actually,
for yourself and for others. That's how I look at it.
Yeah, for sure. I'm actually really glad that you brought up that term herd immunity. Like, do you mind just describing that?
Yeah, so it's kind of like there's a number of people that you have to be immune in order for like those who are not immune to be okay and not become infected. And so I think the number is like 70% or so. So if you have like all these dots that are protected, the chances of that one person in all these protected dots to get infected is really, really low, right? So they're like a herd that's protecting that one person who, may be at risk because the virus can't pass on because no one is getting it in that area.
Got it.
God forbid that one person just happened to get infected, then, that's a different story. But the probability of them getting infected is super low.
Yeah. Oh my gosh, that makes so much more sense now. I've been hearing that term so much, and I kind of had an idea of what it probably meant, but thank you so much for breaking that down for us. I had a quick question, actually, or kind of 2 related, well, maybe not related questions, but one, I wanted to know, you said at the beginning of your presentation that this was an RNA vaccine and that like that's something that we've never really seen before. Like, why are they trying that now instead of doing it like the same old way?
I think the scalability of this vaccine. Yeah. So we're looking for something that can be reproduced really quickly, not that expensive. So you don't have to buy like, things to propagate, the vaccine in the laboratory. And, sometimes that takes weeks and months just to get a certain amount of material that you need. And I think like looking at, previous studies that were done, I think they tried it with Ebola. And I think what happened was like Ebola didn't really become, I would say, a pandemic. So it's, it's not something that they've been distributing, but I think they're looking at RNA now for like vaccines to be developed for like pandemics or things that are happening in the moment. So they've like worked on it before and it seems to be effective, like I mentioned, in the lab and then like the smaller studies that they've done. So I'm hoping that, it transcends to larger studies.
Yeah, absolutely. And that seems to relate to this idea, like the scalability seems to relate to like the light speed type of situation. And just like from an expert like you, like, do you think like speeding up this process, is that something that we should be concerned about? Or
yeah, that's a good
how do you feel?
question. So even as a scientist myself, and I think it also depends on like, who you listen to, like what you watch on the news, and it seems to be like a push on like certain sides. And like you start second guessing, like, is this really like a push for humanity or a push for something else? But what gave me a little bit of comfort was that we saw what happened when it didn't work out. Like the data is public, right? They announce when they have like an adverse event, like when they stop a couple of the studies because of these neurological effects, right? So they're still not carrying on when something happens. They're actually like paying attention to these things. I think if like all went well and we haven't heard anything and it was like, oh yeah, it's smooth sailing, then I'll be like, but I think because it's like the data is public, and I know a lot of times it takes a lot for us to like find the data and then to understand the data, which is like, a whole nother story. But I think the types of vaccines that they're producing, I'm not too worried in terms of like, making it happen quicker. I think there's also a lot more money now for it. And I think money was an issue in terms of like producing vaccines back in the day. It's not a rush, right? So you don't need like $1 billion today to do it. So you get like increments of money and, in a lab, usually now I think everyone is like full force into it. So you have more people working on this one thing. And so I think that also deals with how much shorter like the time frame will be alongside like, there's more money, there's more people, and there's a lot more technology now to do that.
Awesome. Thanks for your perspective on that. I really appreciate it. All right, we've got another question. This one's from Barton on Zoom, and they want to know, there are several vaccines in trial. Once approved, how will we know which one we should take, and will people have a choice?
That's a good question.
That's a really great question.
Yeah, I don't really know the answer to that question because I also have that question and I haven't really done a deep dive on figuring out what the answer is. Because I know they're starting to do some distribution here in the US to like, healthcare providers. And I honestly don't know like which vaccine it is. I don't know if we'll be given the option. Yeah, that's a really good question because they're like, I want to say there are like 30 candidates out there right now. And there are a few that are in phase 3 trials. So yeah, I don't know the answer to that question. That's a really good question. And I'm actually going to look it up.
Yeah. That's really interesting. Yeah, I wouldn't have thought of that too. I actually didn't realize that there were multiple like flu vaccines until recently. And my grandmother told me that she needed the senior one. And I was like, there's another one.
And I think that's probably how they're going to do it. Like what vaccine may be best for like who you are as a person. Like even with the flu, like there's the Flonase one for people who are like allergic to eggs, right? So they have like these different types of vaccine based on, what that person have or underlying conditions. So I don't know if it'll be something like that. And I don't know if you'll have the option to pick and choose which one. That's a really good question. I don't know.
Yeah, awesome. There's another question in from Hello on Zoom, and they want to know, if we only test vaccines on healthy people, how do we determine the benefit and risk ratio for the general population, including those with pre-existing conditions?
Yeah, that is a good question. And I'm not sure I have the answer to that question.
That's okay.
Even our friend, he was trying to do the phase 3, and he went in, and I think he had a high blood pressure, and they were really trying because he's also a Black person, and they're like, we need more Black people to be involved in the trial, but they're like, if you have a high blood pressure, you can't take part. And so I don't know what that would look like if you're distributing this vaccine to people. If they would have to be healthy people, if it will have, is there another option for people with underlying conditions? I don't know. Yeah.
Yeah, we'll have to see.
I doubt these companies don't want to take that risk of testing their product on people that have underlying conditions that opens them up to all types of,
Yeah.
Yeah, that's another tricky situation.
Yeah, it's very tricky. I don't know.
Yeah. All right. Well, on to our very last question. Leslie wants to know, or Leslie is saying, I read today that we will still need to wear our masks and social distance even after getting vaccinated because of relatively low efficacy.
Yeah, so that's what I was mentioning earlier. Like if we're aiming for 50%, we'll still need to take all the precautionary measures that we're doing.
Yeah.
I think we'll still have to wear the mask. I think we'll still have to try to do the social distancing. I think maybe the numbers of people in a certain area may be able to increase if we have a vaccine out there. But like I mentioned, there's many iterations of the vaccine. So I'm hoping in some time, because it's not even also like, once the vaccine is out, and you distribute it, it's also like who's actually going to get the vaccine, right? Who's willing to, take the vaccine.
They're probably telling you you'll still have to wear a mask and everything, more to protect like anti-vaxxers who refuse to take it.
Yeah. So it's like you have to do. I think we're at the point where like let's just do everything that we can to try to significantly reduce the rate of infection.
Definitely.
I think it's going to be just another one on there. And I don't know if next year would look normal. I'm thinking maybe 2-3 years probably till we can, be back to where we were, whatever that was, because I don't even remember.
I know, right? It's okay. At this point, I'm getting very good at matching my masks to my daily outfits.
It's not like a fashion trend. Now I'm like, I'm wearing this outfit today. I'm going to wear this one.
Perfect.
Just take it up and just make it into something great. I feel like we spend a lot of time thinking about the downside, and I think it's really important for us to be, cautiously optimistic and doing what we can, as humans to make sure that we're putting effort into making sure that we can go back to some type of normalcy. And if that means wearing masks, if you go into the grocery store, and then you take it out when you're going outside for a run, then that, right? And, hopefully we can, fight this together. It's not like one person doing it. I think it will take us as a community to do so.
Yeah, absolutely. And you're right. It is easy to get overwhelmed and think about the negative sides of things, but there's still so much to enjoy and so much to appreciate. And I think in some ways, COVID has also made me really appreciate things that maybe I hadn't paid attention to in a long time, like being really intentional about caring for other people in my life and meeting them in a safe way that makes them feel comfortable, whether it be like virtually or like a football field away. Yeah, absolutely.
Yeah. So I think that the goal is just like do things, just do it safely.
Definitely. Oh, and actually, you know what? We just got one more question in. So I'm going to go ahead and ask you one more question, if that's okay. This one says, COVID is affecting people from different racial backgrounds differently. And if the vaccines work differently in people from different racial backgrounds, or do they work differently in people from different racial backgrounds?
That's a hard question. So I think it affects people differently, not because of anything genetic, but more so because of how we live and other things. Like if we think about, how people are treated and access to healthcare, and things like that. I haven't seen any, there are certain aspects in different groups of people that are different in terms of like how we take up certain things and proteins and things like that. I don't know if they're anything based on vaccines, but I think that's why the goal for this phase 3 study is to include as many as we can. And I think they did a really good job of getting that 30 I think it's like 30% or 35%, where like after this phase three study, we can have a better idea of like how this vaccine operates in a large group of different people. So I'm hoping that would look the same.
Yeah, absolutely. No, of course, the ideal scenario is to have everybody that's going, like some representative of every type of person that could get the vaccine in the trial stage, right? But that's obviously a crazy ideal scenario.
Yeah, because we are slightly different. We're a lot the same, but you know,
like the way I take it is a lot of people look at it and they say that the virus is affecting Hispanics and Blacks, and because of their race. It's not just because of your race, it's because the diet and way of life of each race is different. So, you know, Hispanics and Blacks eat a lot, very hearty and very seasoned and a lot differently, and it affects your blood pressure and everything. You know? And that in turn causes
different
effects,
things. You know? So it's a lot of moving parts.
Just because that person is always.
Yeah, that makes a lot of sense. Awesome. All right. So we've made it to the end of our Q&A. I want to take one more quick moment to remind our trivia winners to reach out to us, your Peers and Pub hosts, on either our social media channels, our Taste of Science social media channels, or in the chat box. Send us a personal message and we'll get the details from you that we need in order to send you your prize. All right. And real quick, before you leave us, Odelis, let's take it back to the peer review paper publishing topic for just a second. We know that most scientists have a story about some sort of crazy experience with paper publishing or something like that. Have you got anything to share with us about peer review?
This peer-reviewed paper that I've been trying to write for going on 2 years now. Because life, but also it takes a very long time. Like there's a lot of like minor revisions that have to take place, and you have to like set a time to like get it all done, and thinking about like, who do I publish this paper with? And, you know, is it a right match? What type of figures? Should I go back to the bench and do something else? So yeah, I think for me, it's almost like, I don't know what I'm doing. That's my experience. So which is why I've like also transitioned more into like education and science communication, where I don't have to like spend so much time on like what am I doing?
Yeah, absolutely. Absolutely. All right. Well, this concludes our program. So thank you, Teyvon and Odelis, for joining us. And thank you so much for that informative presentation and both of your inputs for our question and answer session. I just really appreciate it. You guys are awesome.
You guys are awesome. Thanks for having us.
Ah, shucks. Thank you. All right. So I also want to take a moment to thank all of you who joined us from home. We hope you enjoyed our program. And before you go, there is a link to a short survey in the chat available to you. It's just so that you can tell us what you thought of today's event. We want to make sure that we're incorporating your feedback to make this program something that you'll want to attend in the future when we do the next paper. And we really want to make sure that we're making stuff that you like. So let us know what you thought in that survey. And last but not least, make sure you keep up with us, Peers and Pubs, via our Taste of Science social media channels on Twitter, Instagram, and Facebook. And of course, on our website at peersandpubs.org. We look forward to seeing you all again soon. Have a great night. Bye, Odelis and Teyvon.
Good night.